Assalamualaikum and hi, my name is Nur Erysha Ezlyn binti Ghazali and I'm from AS2552B. In this video, I will present about the usage of bioinformatics in conservation. So enjoy the video. Throughout history, natural changes in the environment have contributed to the extinction of flora and fauna. However, disturbance created by humans is amplifying natural extinction rates, reducing plant and animal populations worldwide. As a result, it is important that efforts are made to protect and conserve our planet's biodiversity. For your information, biodiversity conservation is greatly dependent on successfully monitoring species distribution and population size. Traditional monitoring techniques like surveying, species richness, and abundance rely on the physical capture and identification of species. These traditional methods can be hampered by taxonomic identification, but they are invasive, time-consuming, and prone to human errors. Some of these methods make it impossible to detect small and elusive species, so there is urgent need for alternative and efficient techniques for large-scale biodiversity monitoring. The alternative in bioinformatics technique, which called environmental DNA or eDNA, which is increasingly being used in biodiversity surveys and for monitoring. So, what is eDNA? eDNA is the process of extracting DNA samples from water, soil, snow or air without distinctly identifying any particular organism. Species can be classified and identified from the collected samples after they undergo the process of preservation, extraction, amplification, sequencing, and categorization. eDNA has been applied and used to answer questions not only in conservation biology but paleontology and ecology as well. Species can be detected using either eDNA metabarcoding, where multiple species can be identified all at once, or by using quantitative PCR methods for single species detection. eDNA is a simpler sampling approach which can be carried out in most weather conditions. It has proven to be the better method in species detection because it is a non-disruptive method which doesn't cause any harm to the species or its habitat. Elusive and rare species which can sometimes be impossible to identify physically in the field can be identified using eDNA. It also facilitates the early detection of invasive species. Additionally, eDNA enables researchers to observe species occurring in low densities which are somewhat endangered or thought to be extinct. So now I want to talk about DNA barcoding. DNA barcodes are now also being used for the reliable identification and detection of illegally traded and often endangered species. For example, a research team also used DNA barcoding technology to identify shark fins and mantle gills sold commercially and found that a majority of samples they tested from sellers in multiple countries were from a threatened species. In conclusion, bioinformatic tools can be used to analyze molecular genetics data like DNA or protein sequence. Furthermore, conservation of wild species is vital in ensuring the sustainability of biodiversity. In this era of genomics or era of science technology, bioinformatics and the application of computational and statistical tools play a major role in implementing conservation strategies. I think that's all from me. Thank you for watching this video. Bye!